Hello, my name is Michelle Pelchat, and we'll be talking about the Unit 4 case studies. First case study that I chose was the case 1 about the newborn girl that appeared normal at birth, but within 24 hours developed lethargy, hypothermia, and apnea, etc. So the first part of the question is asking about the normal mechanisms of ammonia transfer. There's 4 different, as you can see. The first one that we'll talk about is transamination. That requires the cofactor enzyme pyridoxal phosphate, PLP. And that helps in order to transfer the nitrogen from one amino acid into an alternative keto acid. So during the transamination, no nitrogen is actually released. It is only transferred from one to the other. So alanine, for example, can undergo transamination with alpha-ketoglutarate. The resulting products are alpha-ketoglutarate with the addition of the nitrogen, which is known as glutamate, and then the other side is also pyruvate with the loss of the nitrogen. For the second one is glutamine synthetase. So that primarily functions within the peripheral tissues as a way of ammonia detoxification. So if there's too much ammonia in the peripheral tissues, then that ammonia can then be used to be fixed onto the glutamate that we just generated. And with the use of glutamine synthetase, so as you see here, additional nitrogen added to glutamate, which then results in glutamine. Glutamine can then travel through the circulatory system into the liver. Glutaminase is primarily active in the liver and responsible for the deamination of glutamine. So this reaction frees the ammonia, so the opposite of basically what just happened, from the glutamine, which can then, the nitrogen that was free can enter the urea cycle, and the resulting product is glutamate, which can then undergo a reaction with glutamate dehydrogenase as seen on the bottom. This is a reversible reaction to alpha-ketoglutarate and ammonia within the liver. So it also produces NADPH as seen here. It's allosterically regulated by ADP, ATP, GP, and NADH. So the alpha-ketoglutarate that's resulted can then be used in the TCA cycle, while the ammonia can be used in urea production. So to summarize what we just talked about, you've got glutaminate in most tissues, undergoes a reaction with glutamine synthetase to produce glutamine. Glutamine will then go into the liver, where it has a reaction with glutaminase to produce glutaminate. Glutaminate frees nitrogen to go into the urea cycle. And from here, we can then, that leaves us with glutaminate in the liver, and the glutaminate can then undergo a reaction by glutaminate dehydrogenase, which frees the remaining ammonium, and that will also enter the urea cycle, and we're left with the keto acid alpha-ketoglutarate, as noted here. That can either enter the TCA cycle or can be used as substrate for glucose synthesis or fatty acid synthesis, depending on if we're in the fasted or fed state. Over here in the muscle, we have amino acids. We also have the alanine glucose cycle, which glutaminate undergoes transamination to produce alanine. And then the alanine can be shuttled into the liver, where it can then undergo transamination again with alpha-ketoglutarate to then produce pyruvate. The pyruvate can then enter the TCA cycle, or it can be used to produce glucose through gluconeogenesis and be shuttled back into the muscle to be used as energy. So nitrogen transport is connected to gluconeogenesis, as we just discussed. In the fasted state, protein catabolism is stimulated by cortisol, like we learned in week 4. The free amino acids from this protein catabolism are then used as substrates right here for gluconeogenesis due to the low levels of glucose in the tissues. So you get amino acids that can enter the TCA cycle, generate pyruvate. You also have alanine and other amino acids that can generate pyruvate that we just discussed. Pyruvate can then go into gluconeogenesis to produce glucose, to then use glucose into the peripheral tissues. So in this individual, it was also noted that the orotic levels were high. And so in order to figure out which enzymes are most likely deficient, what least likely to deficient, here's a picture of the urea cycle. So when urine orotic levels are high, that's indicative of high levels of the carbamyl phosphate, which is right here. Carbamyl phosphate is produced by carbamyl phosphate synthetase 1. So if we have excess carbamyl phosphate, it is likely that carbamyl phosphate synthetase 1 is active and functioning well. Now, in order to get a buildup of carbamyl phosphate, it is likely that the ornithine is not combining with carbamyl phosphate via ornithine transcarbamylase, OTC, to then produce citrulline. So if we are not producing citrulline, then we likely have an excess of carbamyl phosphate, which would then be shunted into the pyrimidine synthesis, which would then produce the high orotic acid levels. Therefore, it's most likely that the deficiency is in ornithine the OTC, and we don't have a deficiency here, and we most likely don't have any deficiencies in the downstream enzymes, such as arginocyanate synthetase or arginosuccinate or arginase, due to the buildup of carbamyl phosphate and not the excess of the rest of these substrates. So most likely deficient, OTC. If OTC is indeed deficient or impaired, then that could lead to a decrease in production of citrulline. Citrulline is needed for the urea cycle in order to rid the ammonia from the body, so that could make citrulline then an essential amino acid that we need to get in through our diet, so this way the urea cycle can be completed and we can prevent toxicity in our body. Second case is case 4, about a 58-year-old woman with a bunch of symptoms as noted here. Megablastic anemia is expected. So first off, megablastic anemia is a large group of anemias that are characterized by the presence of larger red blood cells as seen here. What happens here is it's a non-parallel maturation between the nucleus and the cytoplasm of the red blood cell due to the DNA synthesis being impaired, leading to these larger cells here. That's often due to deficiencies in B12 or B6, because both of them are involved in DNA synthesis. So out of the following vitamins, iron is least likely because iron does not have a huge part in DNA synthesis, but it is a very common type of anemia that can be caused in blood loss, problems absorbing iron from the intestinal digestive conditions, or kidney diseases, along with other long-lasting conditions that lead to inflammation. The patient didn't have a lot of these in their history, so also least likely for that. B6 is highly involved in DNA synthesis. The primary role of folate is to donate a methyl group in DNA synthesis. Clinical manifestations are typically without neurological deficits. It's most often associated with mental status change, dermatological symptoms such as facial lesions, glossitis, dry, cracked lips, in addition to the typical anemic findings of fatigue and shortness of breath, which she did have some of these, but the main thing here is that there's typically no neurological symptoms with B6 deficiency. With B12 deficiency, this is where you see your neurological manifestations, with the main symptoms being paresthesias and balance disorders. And clinical exam will usually show a loss of sensation to vibration along with a positive Romberg test. We did see balance deficits in this patient, loss of sensation of vibration in both lower extremities. Therefore, the most likely vitamin deficiency is B12 in this specific case here. So as far as the effect on purine and pyrimidine synthesis here, B12 is a cofactor in the reaction that recycles 5-methyl tetrahydrofolate into the tetrahydrofolate, which is THF, notice here, which is then used to convert homocysteine into methionine. So lack of B12 causes the folate to be trapped in the 5-methyl THF form, which leads to a deficiency in methionine. That donation of the methyl group is crucial for proper DNA synthesis. So if the main storage form of intracellular folate is in the 5-methyl THF, with the inability to generate the THF due to deficiencies in this B12, It's been known to lead to the methylfolate trap. Trapping the folate then leads to a decrease in purine and pyrimidine synthesis, as folate is one of the main substrates for the synthesis of both. The only nucleotide that does not require folate for synthesis is cytosine, as noted in week 15. If there is a B12 deficiency, which affects the conversion of homocysteine to methionine, then there is a noted decrease in methionine available. And methionine is the precursor for the universal methyl donor, S-adenosine methathione, also known as SAM, which is essential for DNA methylation process. And then the last part is what are some potential lab testing findings. First lab test, be a complete blood count to rule out iron. As far as next, you can do plasma serum values. That'll test for B6 and B12. The normal ranges are here. There is the borderline range. So if B6 is considered borderline between 2 to 4, then it's suggested to do urinary excretion of the xanurathic acid. This amino acid catabolites of tryptophan, so this would be done following a measured bolus of tryptophan. Increased levels of this acid would indicate inadequate active B6 for the formation of the amino acid tryptophan. So urinary excretion of xanurathic acid is usually less than 65. So excretion greater than this threshold would indicate abnormal tryptophan metabolism due to a B6 deficiency. If B12 is found to be borderline in the serum values, then it's recommended to do the methylmalonic acid testing, and that can help differentiate between B12 and B6 deficiency, as homocysteine levels are elevated in both B12 and folate deficiencies. And that is also typically serum values, and then if B12 is considered borderline, then we would go to the methylmalonic acid testing to definitively rule in the B12 deficiency and rule out B6. But it's likely that they have both from the research that I found. And that is all.